So ꯑꯩꯈꯣꯏꯁꯤ ꯉꯔꯥꯡ ꯇꯥꯛꯈꯤꯕꯁꯦ some historical background ꯍꯥꯏꯇꯥꯔꯦ photosynthesis ꯍꯥꯏꯕꯁꯦ ꯀꯔꯤꯅꯣ ꯈꯪꯅꯕ ꯍꯣꯠꯅꯕꯗ scientist ꯃꯌꯥꯝ ꯑꯃꯅ ꯇꯧꯈꯤꯕ contributionꯒꯤ ꯋꯥꯔꯤꯁꯤꯡ experiments ꯂꯩꯅꯈ꯭ꯔꯦꯕꯀꯣ ꯉꯁꯤꯗꯒꯤ ꯇꯥꯛꯇꯣꯏꯁꯦ will start from where do photosynthesis occur photosynthesis ꯀꯥꯏꯋꯥꯏ ꯆꯠꯊꯒꯦ ꯉꯁꯤꯒꯤ ꯇꯣꯄꯤꯛꯁꯤ site of photosynthesis ꯍꯥꯏꯇꯥꯔꯦ where do photosynthesis occur ꯁꯤꯅ ꯇꯣꯄꯤꯛ ꯑꯣꯏꯍꯟꯁꯤꯀꯣ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁꯁꯦ ꯀꯗꯥꯏꯋꯥꯏꯗ ꯇꯧꯕꯒꯦ ꯍꯥꯏꯌꯦ ꯑꯣꯀꯦ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁꯁꯦ ꯀꯥꯏꯋꯥꯏꯗ ꯇꯧꯅꯤ ꯄꯥꯝꯕꯤꯒꯤ ꯀꯔꯝꯕ ꯑꯣꯔꯒꯦꯟꯁꯇ ꯔꯨꯠꯇ ꯂꯤꯞꯁꯇ ꯁ꯭ꯇꯦꯝꯗ ꯖꯦꯅꯔꯦꯂꯤ ꯂꯤꯞꯁꯇ ꯇꯧꯋꯦ ꯑꯗꯨꯅ ꯁꯔꯒꯤ ꯏꯍꯥꯟ ꯍꯥꯟꯕ ꯄꯣꯏꯟꯠꯁꯦ ꯃꯨꯟꯅ ꯇꯥꯎꯀꯣ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁ ꯇꯦꯛꯁ ꯄ꯭लेꯁ ꯏꯟ ꯗ ꯂꯤꯞ ꯌꯥꯔꯦꯀꯣ ꯂꯤꯞꯁꯀꯤ ꯀꯥꯏꯗꯅꯣ ꯍꯪꯉꯦꯕ ꯂꯤꯞꯁꯀꯤ ꯃꯦꯁꯣꯐꯤꯜ ꯁꯦꯜꯁ ꯌꯨ ꯔꯤꯃꯦꯝꯕꯔ ꯃꯦꯁꯣꯐꯤꯜ ꯁꯦꯜꯁ upper epidermis and lower epidermis ꯑꯅꯤꯒꯤ ꯃꯔꯛꯇ ꯌꯥꯎꯔꯤꯕ ꯁꯦꯜꯁꯤꯡꯗꯨ ꯃꯦꯁꯣꯐꯤꯜ ꯁꯦꯜ ꯀꯧꯋꯦ ꯍꯥꯏꯗꯅ ꯂꯤꯞꯇ ꯇꯧꯏ ꯂꯤꯞꯀꯤ ꯀꯥꯏꯋꯥꯏꯗ ꯇꯧꯗꯣꯏꯅꯦꯕ ꯂꯤꯞꯀꯤ ꯃꯦꯁꯣꯐꯤꯜ ꯁꯦꯜꯁꯇ ꯇꯧꯋꯦꯕ ꯃꯦꯁꯣꯐꯤꯜ ꯁꯦꯜꯗꯨꯒꯤ ꯀꯥꯏꯋꯥꯏꯗ ꯇꯧꯗꯕ ꯀ꯭ꯂꯣꯔꯣꯄ꯭ꯂꯥꯁꯇ ꯌꯥꯔꯅꯤꯀꯣ inside the chloroplast ꯃꯅꯨꯡꯗ ꯇꯧꯗꯣꯏꯅꯦꯕ ꯑꯗꯨ ꯑꯣꯏꯇꯥꯔꯗꯤ ꯁꯦꯟꯇꯦꯟꯁ ꯑꯃꯈꯛꯇ ꯀꯃꯥꯏ ꯍꯥꯏꯕ ꯌꯥꯏ photosynthesis takes place in the chloroplast of mesophyll cells of leaves ꯌꯥꯔꯅꯤꯀꯣ chloroplast ꯇꯧꯗꯣꯏꯅꯤ mesophyll cell ꯃꯅꯨꯡꯗ ꯌꯥꯎꯕ chloroplast ꯀꯅꯥꯒꯤ mesophyll cell leaves ꯀꯤ leaf ꯑꯅ ꯇꯥꯔꯛꯇꯣꯏꯅꯦ ꯌꯥꯔꯦꯀꯣ ꯑꯗꯨꯅ ꯍꯧꯖꯤꯛ ꯁꯔ ꯑꯩ ꯅꯍꯥꯟ ꯃꯇꯦꯛ ꯑꯃꯇꯪꯗꯤ ꯌꯦꯛꯄ ꯍꯧꯗꯤ ꯍꯧꯈ꯭ꯔꯕꯅꯤ ꯀ꯭ꯂꯣꯔꯣꯄ꯭ꯂꯥꯁꯇꯣ ꯁꯔ ꯑꯝꯇꯪꯒ ꯊꯝꯈꯤꯒꯦꯀꯣ ꯈꯤꯇꯪ ꯃꯨꯟꯅ ꯇꯥꯃꯤꯟꯅꯁꯤꯀꯣ ꯉꯁꯤꯒꯤ ꯄꯣꯏꯟꯠ ꯉꯁꯤꯒꯤꯁꯤꯅꯤꯀꯣ ꯈ꯭ꯋꯥꯏ ꯃꯔꯨ ꯑꯣꯏꯁꯦ ꯍꯌꯦꯡꯗꯩꯗꯤ ꯑꯩꯈꯣꯏ ꯃꯦꯀꯥꯅꯤꯖꯝ ꯇꯥꯛꯄ ꯍꯧꯈꯤꯗ꯭ꯔꯣꯕꯅꯦꯕ ꯃꯦꯀꯥꯅꯤꯖꯝ ꯅꯠꯇꯦꯕ ꯃꯔꯨ ꯑꯣꯏꯁꯦ ꯑꯗꯨꯒ ꯃꯦꯀꯥꯅꯤꯖꯝꯗꯨꯕꯨ ꯈꯪꯅꯕ ꯍꯣꯠꯅꯕꯗ we need to know the apparatus the raw materials ꯀꯔꯝꯕ ꯂꯥꯏꯠ ꯗꯔꯀꯥꯔ ꯑꯣꯏ ꯁꯤꯒꯨꯝꯕ ꯃꯌꯥꯝꯁꯤꯅꯦ ꯑꯗꯨꯅ ꯉꯁꯤꯁꯤꯅ ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯃꯔꯨ ꯑꯣꯏ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁ ꯑꯟꯗꯔꯁꯇꯦꯟ ꯇꯧꯕꯗ ꯌꯦꯡꯉꯣ ꯅꯍꯥꯟꯁꯨ ꯌꯦꯛꯈ꯭ꯔꯕꯅꯤ ꯁꯔ ꯑꯝꯇꯪꯒ ꯌꯦꯛꯂꯛꯈꯤꯕꯀꯣ ꯀ꯭ꯂꯣꯔꯣꯄ꯭ꯂꯥꯁꯇꯣ look here so let's say this is the chloroplast see chloroplast ne lorse chloroplast se koina taakpada chloroplast has got two membrane right mapan thangusina let's suppose this is outer membrane ai outer membrane se OM aina tharage yarabo chloroplast ki outer membrane sina chloroplast ki inner membrane ne yareko nahan du suta kibada chloroplast ki manung se the interior part of the chloroplast is filled with a fluid and we call it stroma haina ko manung sina kai ko ba manung si stroma kawe yareko hairiba stroma sida there are many disc-like structure mayam ama leibu iyu there are many disc-like structure here these disc-like structure are called kai kawe haigiba thylakoid kawe haigi hairiba thylakoid mayam sina maron longkhatnara finally it will forms grena adudi sarna yisi thylakoid ama thylakoid ani thylakoid ahum thylakoid mari thylakoid manga se maron longkhatnare yarniko amamam se namtang level toronggi amamam sidi kai kaweba amamam si thylakoid kawe thylakoid yareko hairiba thylakoid singsna group oina maron longkhatlakanda we call it grena haina kawi singular dana we call it grenum haina kawi adudi aina hairaktuise thylakoid haiba grenum haiba se chap manai ko thylakoid haigana amakhakhtang haiba grenum haina group oina haiba khaktane yarniko hairiba thylakoid sigi membrane da on the membrane of the thylakoid the pigments kari pigments the chlorophyll pigments are found attached nappa layer kai wai dano leise thylakoid ki membrane da hojik sarna sida pink machu mayam hapise kai neba these are the pigments are found located on the thylakoid membrane ai adudi asang machu se antang level toksange ko green machu se kai neba I will simply put it pigments haige sir green oina uram utlabanina aina main oina uliba pigments si chlorophyll pigments de yareko now hojik sarna nahan haiba kanda photosynthesis se is done in how many steps two steps natra photosynthesis taugai de photosynthesis adi panbi na machinjak sungbudo तवतो मचिन जाक तो सुंगेदी कई हाना लेतदी यादे एनर्जी हाना लेतदी यादे अना इहान अनब स्टेप ता एनर्जी हाना लेतब यारोय अदुगी एनर्जी प्रिपेयरिंग स्टेप तो कई कवे हाइगी सरना लाइट रिएक्शन कवे हाइगी अना होरेन सनलाइट से लाकांदा solar energy sunlight se kanana trap tawdano hai tardi solar energy is trapped by the pigments present on the thylakoid membrane thylakoid ki membrane da yauri ba kanana pigments singduna sunlight se trap torni and convert it into usable form of energy called ATP and NADP is to the ontoctone. What is the job of the pigments? The job of the pigment is to trap the light energy, convert it into ATP and NADPH2, which is what we call as light reaction. Why? Because we need light to prepare the ATP and NADPH2. Yareko, what is coming in? Light. When I put ice mu, light is coming in. The incoming light is now trapped by the pigments and convert it into. See, light se trap torokanda kai sungeva. The light energy is finally converted into usable form of energy that is ATP and NADPH2. The on the haineva. Adi where exactly light reaction happens? Light reaction happens on the thylakoid membrane doon sa tape. Once high energy si heck later. Once this energy is available, then what happens? In the stroma, kanaga punsa na bayare, carbon dioxide now combines with, wait, just a second. Minam tama ko makarom doon ige. So, carbon dioxide combines with kanaga combine tawreba water ga and forms what? And forms glucose. Yarnika, high glucose C reactions at Tokai it happens in the presence of this energy. See energy see presence at 12. Yarniko, what energy? ATP and NADPH2 की presence the carbon dioxide and water get fused and form glucose. This particular step is called dark reaction. In a purple na irive steps is in a dark reaction ni, yellow na irive steps in a light reaction. Yareko. Aduna photosynthesis kai way the toki haibedu di khangeva. Ado akoi di photosynthesis na tamaroi. Photosynthesis haibasi light reaction plus. So photosynthesis ki light reaction do kai de to ino in terms of kai da on the thylakoid membrane haigera grena haigera dum chumiko. And the dark reaction happens in the stroma. Aduna horen se ai sir iba kanda sumaina inarkim. For chloroplast sigi chloroplast se chloroplast sir maphamani reason ani thokna thamge thylakoid ki part ama aduga thylakoid yaudaba part jaga singshina kai koeba we call it stroma haina koeba yare ko sik thylakoid or you can say kanagumba kharana grena haina iba yai ko group oina thylakoid ki part ama aduga stroma ki part ama sumain thumsi yare ko mayam hairiba thylakoid sidana kai thoktoneba site where light reaction occurs. Sidana kay ba light reaction toktonem. It is the site where light reaction takes place. Light reaction occur. Light reaction tokpam ni si jaga sina. Whereas stroma sina kay tokpam it is the site where dark reaction tokpam ni sina. Dark reaction occurs. May samjalagya ko. Now, sida question ang kahang bayay. ꯀꯩꯗꯧꯕꯒꯤ ꯁ꯭ꯇ꯭ꯔꯣꯃꯥꯗ ꯗꯥꯔꯛ ꯔꯤꯑꯦꯛꯁꯟ ꯊꯣꯛꯏꯅꯣ ꯍꯥꯏꯅ ꯑꯩ ꯀꯣꯟꯅꯁꯨ ꯄꯟꯗꯤ ꯄꯜꯂꯛꯈꯤꯅꯤꯀꯣ ꯗꯥꯔꯛ ꯔꯤꯑꯦꯛꯁꯟꯁꯦ ꯇꯧꯕ ꯃꯇꯝꯗꯨꯗꯗꯤ ꯄꯜꯂꯛꯀꯦ ꯍꯧꯖꯤꯛ ꯖꯁ ꯃꯃꯤꯡ ꯈꯛꯇꯪꯒꯤ ꯄꯜꯂꯛꯈꯤꯒꯦꯕ ꯗꯥꯔꯛ ꯔꯤꯑꯦꯛꯁꯟꯁꯦ ꯇꯧꯕ ꯃꯇꯝꯗ we need an enzyme called Rubisco ꯍꯥꯏꯅ enzyme ꯑꯃ ꯗꯔꯀꯥꯔ ꯑꯣꯏꯌꯦꯕ and that Rubisco is found in the ꯁ꯭ꯇ꯭ꯔꯣꯃꯥꯗ ꯂꯩꯌꯦꯕ ꯁ꯭ꯇ꯭ꯔꯣꯃꯥꯗ enzymeꯗꯣ ꯐꯪꯉꯕꯅꯤꯅ ꯁ꯭ꯇ꯭ꯔꯣꯃꯥꯗ ꯊꯣꯛꯏꯅꯦꯕ ꯌꯥꯔꯅꯤꯀꯣ like ꯍꯣꯔꯦꯟ ꯁꯔꯅ respiration ꯇꯥꯜꯂꯛꯄ ꯀꯥꯟꯗ Krebs cycle se matrix the thui se kaigino mitochondria gi hack tardi all the enzymes required for Krebs cycle are found available in the matrix hackani except exception ka di laini but normally matrix ta phangbai ne yare ko aduna particular reaction ama se thoke hai tardi kadai wai da reaction se thokani hai tardi reaction du darkar oba enzyme leiba mafam du doina thokali heba yare ko we will come to this kona lakse sibo khamadra sina introductory part ne now hojik hairiba chloroplast kaida yaudana mesophil cells kanagi lips ki aduri sir mesophil cell amta ye kyun ko si question ti hangbani ko swaida ai hanjan hayori sini this is mesophil cell mesophil cell ningdi ningsangi ko mesophil cell kana marakta leiba cells deba upper epidermis ka lower epidermis ka if you remember dichotomy upper epidermis is tightly packed parallel to one another, we call it pellicet, while the lower are loosely arranged, called a spongy mesophyll cogumnal. Whereas in case of monocot, no such differentiation, just mesophyll. Yarage? Adudi hojik sir na yise kai potneba, ye orange machuna yise mesophyll cell name. Adudi lip manung di over cell amanatra. Sigi manung da mukana yaudoneba, chloroplast yaudoneb. Ay hana level ta hogi ge na tari horen prom laitanaba. Sir na sida orange na yise kai cell neba. I am drawing mesophyll cells neba. Yare ko? Ado ay ki level sidi mesophyll cell dugi cell wall na nis. Yare ke ko? Ay cell wall ga I will simply put it mesophyll cell. Cell sigi manung da mo kai yaudoneba. Kai yaudoneba chloroplast yaudoneba. So many chloroplasts. Chloroplast mayam yawe. Yare ko kanano ya ay ni yise chloroplast. Yare ko kanano ay ni yise chloroplast ne. Yare ko hozik ay na harak to ise mesophyll cell ani sida chloroplast ki arrangement yek pasi kitang khenadra. Kamain tawge ham tamda light yam laytaba matamda. Yani ko dim light or low light intensity ki matamda. I'm going to write here low light. Light yam laytaba matamda arayba kharado maximum absorb toba yana hotnada light na laiba direction do chloroplast perpendicularly arranged away. Congress na say light si kitang laeba light si maximum wina raw gi tare ay halgo laita light si smile ay na smile lepe perpendicular light si am laksa leba ado tare ay smile lepe parallel win check na yani ko kay gino da chlorophyll pigments the lights directly yay tare gadi chlorophyll pigments in the damage the photo oxidation toroga yam ye hen da suya de yani ko ana ka hena laksalaba highlight in the numit chap yung matam dadi chloroplast sing they are aligned parallel to the direction of light to minimize okay, maximum exposure do adu handana minimize to na parallel when against the whereas during low light intensity matam dana light to yam lahu ya na perpendicular when against the badare yare ko adu di si karam kanda low light to thokpa arrangement alignment never sina aduga high light intensity ki matam da high light yani ko or bright light ki matam da di kaidoge ham tamda light na laiba direction du da chloroplast ki alignment se parallel oina leini khangro sarna haining ise khangi ko adu noi khanga da mai kanam ta haige ko photosynthesis kwai waina maximum to du shi numit chap yung bu da nati ko yani ko photosynthesis kwai am na to du se early morning and adu horen numit taram dai matam sidane yani ko pung 8 ga swai tar ko sida maximum to we hai adu nuktin pung 3 mari ga tar ko swai yam nga la su to de have sarna ise kaigino da maximum sunlight intensity yang wang tar de aduna thom badane chlorophyll pigment se kaido eba photo oxidized or damaged to eba aduna ai ngale har kini aduna chlorophyll pigment se oxidized to dana to pigments ram na yom sen bi layer kani har ani ko we'll be talking about that in photosystem ko topic ang manung da tala kini aduna sir sig arrangement sim tanga palam gi ko swaida kai pando ba sumaina alignment of chloroplast chloroplast se kamaina align tauge haibase ecosystem do paraga layuko do haidi sarna si khatang takpani na ko horen halla si khaktudi pungkhai am da loi khani ba pa ba ana adum kanda ecosystem ra khar focus to wiroi si di nang na ba dum tala ke ba ko chloroplast se kamaina align tauge haibase how the chloroplasts align themselves that depends on the kana depend on the intensity of the light that depend there yani ko alignment of the chloroplast depends on kanada depend however depends on intensity of light or I should say light intensity that depend however kamaina arrange tawi low light intensity matamdana direction of the light to the kedodawa perpendicular oina leidoba highlight ki matamdana parallel oina leidoba adumaina thamse ko sir aduna condition se ani khaidoroke during low light ki matamda kamain tawi haibama aduga highlight ki matamdana kamain tawi haibama high light intensity ki matamdana yare ko low light ki matamda how the chloroplasts align themselves how yani ko chloroplast se kamaina align taweba chloroplast align ay tension ire ko align themselves parallel or perpendicular, parallel to nattava, perpendicular to direction of light. Light na laiba maikay do direct kani direction of light. Sumain thamage asom side to na tensalage. Sidana chloroplasts align themselves kamaina parallel to light. Light ka parallel to light. Why? Minimize tong maximum exposure later. Yarake ko sina. So this is all about kanagi wari neba sina photosynthesis kadai waida tawi haibadugi wari. Yarni ko and a little bit of chloroplast structure galay. Sigo yara sira. Can I change the slide? Slide change toroge ko. Next. Mathangda raw materials required for photosynthesis. Photosynthesis to matamda raw materials kari darkar oige se. Direct NCRT da yaude ay yaubadu khaktang neba notes when idoidi. But tam da badi yadaba. Yareko aduna aikoigi mathanggi topic se raw materials. Somda kitang muna tabiu. Sir amta skip tawroi somda yengbiu. Raw materials for photosynthesis. Photosynthesis tawwa matamda sir se ai noina tharappa message khara ai hanba ngamdri kitang ngaiko ko ai isa phatri yam sole hor nungdang ga loidoni revision gal work matamdri yam ti sole raw materials required for photosynthesis. Photosynthesis tawwa matamda kari raw materials darkar oige amta haibiraksi. Number 1, yes, carbon dioxide, water, sunlight, pigments. Yareko. So these are the raw materials required for photosynthesis. And the photosynthesis the darkar oiba raw materials sir amtang han ithoge ko. Ahan the first raw material is kananeba carbon dioxide ne. Sidai kem hiding ai late. But just general knowledge when ko. Terrestrial plants study carbon dioxide se kai de lower from the atmosphere itself in the form of in carbon dioxide gas ki form to the lower. Whereas aquatic plants study carbon dioxide se bicarbonate ki form to the lower. Dissolved form. Kangi ko aduma haipetang idea le view. Ani subase kai neba. The second raw material is water. Yare ko ngarang khangmi nagre sarga. The role of water is to act as reducing agent when ectowave. Oxygen released during photosynthesis comes from water and not from carbon dioxide. Yare ko water ki role se kai donor no ma lauri sa hydrogen donor oina lauri ne heba. And even without water, can there be photosynthesis? Yes, if we have the reducing agent. Hydrogen donor do laire ti tum yae. Yare ko ahum suba raw material se the third raw material is sunlight. I should say light. Yarni ko matang swaida light ki any wavelength see photosynthesis the use the way any form of light se photosynthesis the use the way abradi yadi. Karamba light no photosynthesis the use to doishe haitardi visible spectrums of white light. Yarni ko high violet, indigo, blue, green, yellow, orange, red. Sigi wavelength se khatane photosynthesis se. Shorter wavelength and longer wavelength are not suitable for photosynthesis. And the karamba light say photosynthesis the use the weva visible spectrum, the visible light I should say visible light, or we can write white lights in it. Photosynthesis the use the say white light. Karamba light ki wavelength ranges from about approximately 400 nanometer to 700 nanometer. Swagi range in it. Yarni ko kadai buki range in it about 400 nanometer wavelength of light to 700 nanometer wavelength of light. I NCRT da sumaina hapet P haina iramni. Aseng badi 380 nanometer to 760 nanometer swai buk se yabani. Yare ko Kaniko, Sidai Shibuki wavelength Shibukanano, it is none other than violet, indigo, blue, green, yellow, orange, red mapasnayo. Adi 400 to kanawaram tare, violet or amtare, 700 to kanawaram tare, red or amtare. Yareko, Basima Kaminarsi. Violet ay 10 bam tahirona. Wavelength. 10 thagi basing. Sidagi atin bama, awaang bama. We have heard about infrared, right? Moiduna sida awang bakambuneva. Sida badana we have the UV rays, the X rays, they are not suitable for photosynthesis. The plant cannot use those radiation. Yarabo, yareko. Which one is having highest energy? Which one is having highest energy? Violet. Violet, Nico. Longer the wavelength, smaller is the frequency, right? Frequency PAD energy ke dorne. Energy name equal to S mu do. Yare. Yare ko sima. Ahum mari subase. To C neva. Yawisina. The fourth raw materials that we required for photosynthesis is none other than pigments. Yarabo, kay darkar weba, pigments darkar web. Kitang muna tabi siko, na inakangdaba kara chongthonak. Photosynthesis tawada, as written in the NCRT da ibe matungina pigments makal ahum darkar way. Yareko, kari pigments darkar ay adum three types hay ka. We require as many as three types of pigments. Pigments makal ahum darkar oy. Pigment makal ahum sida mahanba pigments se. The first one is the most important one is chlorophyll. Mahanba pigments se kay chlorophyll kawi. Yareko. Chlorophyll se NCRT matungina makal ani tong kay Ado chlorophyll makhala ni khata nate ko yam lei chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D na hawai su tam giduni kaida a plant kingdom da. Yare ko adubu sigi chapter sigi matuina higher plants da ma chlorophyll se ani thong thame. One is chlorophyll A, another is chlorophyll B. Yare ko ani suba pigment se the second pigment is called xanthophyll. Haine ko. Kai ko ba the second pigment is xanthophyll. And the third pigment is carotenoids hay se. Kay ko ba? Carotenoids. Yaribra? Yare ka? Pigments haibase kaino color piba ngamba substance ne. Aduri mo si machu machu lo oiramgani e. Chlorophyll A gi machu sa. Direct ibe matungina bright or blue green machu e. Kay machu e ba? Bright green or blue green. Oy gani. Yare ko hanadagi test yam tharapa kambu khamani si color si the best of question je adum sahi guding hangis. Test yam hangye base. NEET hangye am nate ko. NEET tasu hangba yaugre. Mock test de dum yaurani ma compulsory. Chlorophyll B say kai machu oige it is yellow green way. Chlorophyll boy tari green di yaurni ko. Ada shina kai machu oiba yellow green. Sidi khada ba yaroi ko. Haibana yaroi. Yare ko. Xanthophyll say ma se kai machu oige di yellow machu oe mana. Kai machu oiba it is yellow in color. Whereas carotenoids say Yareka, my machusna yellow to yellow orange web. Yellow the gina, kitang yakshan ba orange pobo obi henny. Yellow to yellow orange color obi. Kamai kangai no machu meam set. Hyperkanda, panbi de yori ba pigments makal hoima broadly home, ado ako Maria Harsun ko edus ega bigas tongana. Hairiba machu makal mari set. Koi kamai kanga ke hatari by doing a technique called pepper chromatography, haina technique ama torokanda kangyo. Yareko, I'll show you what is pepper chromatography. Pepper chromatography hai se kem nate. Noi noi di MBBS tam drom ni na pepper chromatography tam maroi. Masida. Yareko, graduation low tarya. Pepper am khak ne bako. Sigi pepper se sidai sida ko na lip ama panbi ki green part am lower se. Especially lip lower ge. Ram the pigment se lip to yaw do ni na ka. Lip se ko na takhai re na. Mahi ola le na. Mahi sida thada ba pepper sida. For example, si jaga se thada rage. Masai mahi do. Lip to gi mahido ko khairaga khairakan thadaba kanda mangonda yauramba pigment singse separate on the paper adugi hojiki paper chromatography paper sida separate tori yani ko kamaina separate torke da si kanano amta haibiro paper sida yellow machu jaga sing spot myam ure yellow di kanar mane xanthophyll ne yellow to yellow orange amtang thamge ko yellow to yellow orange orange kitang sangchan doga si kanano sina carotenoids ne. Yareko blue green adudi okay. Blue green machuse kanano this is chlorophyll A. Yareko madukai yellow green yellow kitang oyan gedudi okay. This is yellow green. Sina kana Sumain toroga on the chromatography paper to the we can separate out the pigments. Okay? Okay? When they are separated, they appear different colors. Urieva. Aduki color do ne akoina shida hapise. Yareko. Adu tardi, this paper is showing different pigments with different colors, right? You know, often toriva different pigments we call it chromatogram. Is it no? The technique is paper chromatography, and what you obtain in the paper chromatography is called kay paper chromatogram ko. Si ay na uli siya kayo no? Chromatogram. Bro, how is chromatogram obtained? By doing a technical chromatography. Hariba technique sa general. Yam ka na darkar wede, hapetang ay kangba nungay na ba na hapsalamani. Si kayo na. Blue green. This is representing chlorophyll A. Okay, what is this? Kaino say yellow green chlorophyll B. What is yellow? This is xanthophyll. Yareko, what is this? Yellow to yellow orange? This is carotenoids now. Yanina. And the entire pepper showing different colors is chromatogram. Hanako. Yarage. Yareko. Ay, hapetang ningsang kenway ningsang you remember pollen teeth? Pollen teeth. Ningsang ka. What is the color of pollen grains? Yellow iti, yellow iti. Pollen grains yellow iti normally whitish in color oyebuko. Ado yellow iti do kaygino, ado yung mathakta mo white yellowish sticky substance na cover to yun. Characteristic features of only insect pollinated flowers ni haygibdo. That sticky yellowish substance is called pollen kit ko. Yare ko, kaygino sticky o halamide horen koydon la tatlong na pinalo. Insect pollination da mating pang na banib. Yare ko, Adudo, no ningsang am gidra di khangde NCERT today the pollen kit is made up of lipids and carotenoids. Haygino. Adun ne kay machuido. To because carotenoid is yellow to yellow orange soido. Yare ke sibogta. Yare ko. Uh, carrot ta kayawi. Carotene ya. Kay machuoy. To aduna yellow to yellow orange so nido mai wena phabiyo carotenoids ki matang de. Sima ay khangi haina loroge. So these are all about the raw materials. Can I change the slide? Next. Matangda ay ngarang hapetang di palamam ni ko. Let's recall few things about absorption spectrum and action spectrum in details na inadrom ni Jose. Yare ko, Jose, ani kaktang yingisi term ani kak one pole amana kay ko ba? Amana absorption spectrum ama. Who mentioned this two term? T.W. Engelman. Very good. He did the experiment using which plant specimen? Cladophora, Sienna, Tohi. Yare ko, one is absorption spectrum, Chumbra. The other one is action spectrum. Ay ani sa dong comparison tawge ko, iyeng ba nungay na ba? Action spectrum. Okay. Absorption spectrum ga action spectrum ga se kaino matang sida sir na ibya sa anima graph ngaktanew. Okay? Absorption spectrum is a graph. Karamba graph no haibaduri ngahak lerga makha itharakshi. Action spectrum sisu graph ne. Yare ko. Absorption spectrum sina ngarang pandi na khwaidagi pamjana absorb tawgiba wetland do karamba na. Karamba pigment na karamba wetland of light se absorb tawi haina takpa graph to. We call it kay ko ba absorption spectrum. I will show you here. Ulati yung ko. Graphically so tawseba adong ganda si wede na hayag to ni. Okay, look here. So on the x-axis, I'm going to put the wetland. Som side kay thamaseba wetland thamaseba. Yani ko. And on the y-axis, I'm going to put rate of absorption. Chupsen de kisa rate of absorption. Wetland say 400 to 700 thamdoni. Ado masing wena ibay mo ta color wena irase do nalayani. Violet, indigo, blue, green, yellow, orange, red. Thamage sir, sumayin thambage sukay de ko Violet 400 nanometer, about 450 more already indigo money, about 500 maya or the blue to sumina, violet, indigo, blue, green, yellow, orange, red. Thameko. Peshukai the rose color win at Thambase. Okay. Hozik, akoi absorption spectrum kaino yese. Chlorophyll kanase. Chlorophyll A se. Chlorophyll A hariba pigments in a quite palms and absorb the pigments there. Blue and the red jagasine. Kanana. Chlorophyll A na khwaidagi pamjana absorb tawse kanai wari neba sarna irise sida this is for chlorophyll A pigments. The pigment called chlorophyll A absorb maximally at two regions, one at the blue, another at the red region they absorb tawe. Chlorophyll B gi oinadi yare ko chlorophyll B gi oinadi kadai waida absorb tawi heba kanda magi absorption sina little away from blue and little away from red. Red tagi kitang kitang. Adom red maya aduni ko. Ada kitang kak. Yare ko. Sina kanagi wari neva. Sina chlorophyll B gi wari neva. This is for chlorophyll B. Whereas carotenoids na pamjana chupsava jaga sina kadai wai noite somewhere between the blue and the red jaga wai da. Sina kana carotenoids kine. Ay shumaina dum ye ke ko larka kitang mana draso. Fara loire koi da ikamai phado no harake. So what do you know from the graph? Karamba pigment na karamba wavelength of light absorbed the weak. Kung mga graphs lagi. So those graphs showing which pigment is absorbing which wavelength of light is called kay koiba absorption spectrum koiba. Graphs lagi pwede na kung bada maruin na chlorophyll na quite absorb to this somewhere the blue region and somewhere at the red region. A wagera B wagera blue mapa red mapa swai da loy throw. Ay tari quite maximally absorb to the jaga si blue mapa ga red mapa ga ne. Yarni ko how do we dear target absorption spectrum se combine ibatai? It is a graph showing karamba graphs in a sort of this kaina. Graph showing different pigments absorbing different wavelength of light. Tongan ba pigments na tongan ba wavelength of light absorb tawi haina utpa graph to. Madu bo koina kay ko ba absorption spectrum showing different pigments absorbing kay different wavelength of light absorbing different wavelength of light. Yare hairo. Yarabo. Adoi tare panbina khwaidagi pamna lokshangiba wavelength se blue mapadama red mapadam lokshan grena. Light am lokshali mapa se kai jaga neba blue ga red ka. Light am lokshan dari yam lokshan bam jaga do yam photosynthesis tormani. Yare ko. In terms of photosynthesis the hierogana action spectrum go. Yani ko I will change it. Kitang change to throw kyo ka. Aduga gyan tabra amuk engmina seba serga. Like this. Sumaina. Chap manay kenariba haibabu ko. X-axis is ako'y itong wavelength thamsi violet, indigo, blue, green, yellow, orange, red. Violet, indigo, blue, green, yellow, orange, red. This side is representing the wavelength. And on the y-axis, I will change it as kay rate of kana onto se wandaktia photosynthesis. Handa mukti photosynthesis ke rate onto hoge. Yani ko sumaina yekpa matamda graph se sumain taeba. Kai jaga da photosynthesis quite wang hai na. The blue ga red ka. Why? Aswai jaga da na quite subsan gi neba. Since absorption spectrum is highest at the blue and the red region, so obviously the rate of photosynthesis kai weda. Yam lokshan gram ni na yam lokshan ban jaga da yam action torone kai action na torisa. Photosynthesis hai riba action ji uthore. Yarni ko aduna shigi graphs na kai ko eba action spectrum go. Adi how will we define graph showing rate of photosynthesis at different wavelength of light. Yani ko, tungan ba wavelength of light sing da photosynthesis karam na tawrige takpa graph to na action spectrum ko at different wavelength of light. Hangi bro sarna pa ise Hozik somsina kai action spectrum somsina kai absorption spectrum action spectrum ga absorption spectrum da pairaga thonjana matamda almost overlap toyo maramdi si jaga se kai jaga dan khwai wangi luga si su kai jaga dan khwai wangi the graph almost overlap right superimposed over exactly di nate ko maramdi carotenoids ki role khara su leiri and exactly superimposed away the high gumsi the two graph that is the graph for action spectrum and the absorption spectrum almost overlap the Weber graphs. Yarabro Sibokta. So this is all about the two spectrum, absorption spectrum, the action spectrum. Can I synthesize? Next. Now let's talk about photosystem. Hodrom ni ko. Sina aroi ba topic ko idoni. Ngasay morning dali takpan ng di, noy da takpan ng doman. Now, matang gisi. Som nam tayeng bisi. Hojik sir na talk to about topic si maming photosystem. Okay. So tami na siya. Ngasay photosynthesis tawag matamda, do we require pigments? Yes, we require pigments like chlorophyll A, chlorophyll B, we require carotenoids, xanthophylls, the whatever. And also we know that all these pigments are found located on the thylakoid ki membrane da nappagalay hai na. Thylakoid ki membrane da na hariba pigments singse amamamtang individually napte. They occurs in group oina lay. Yani ko adugi group sing the horen photosystem kawi harak to ne. Aduna sir amtang machi amtang recall torki ka. Chloroplast ki manungda yawa big sep structure singdo kay neba ay thylakoid amtang thadrokep natragana noina nungay tragana ay amtang back chalam gese sila si chloroplast ni ko sar na yekrisa amamam se kay kaweba asi thylakoid se amak khaktang ay thangdogi ko hojik sir kuta pairi asom pe so pora si okay sida sida kay no yekto sida thylakoid chloroplast nate ko ay na yekto si thylakoid let us say this is thylakoid okay Sarni si thylakoid si membrane ani ne heba nate ko yare ko kaigino da membrane haba wai si wai awraba plasma membrane oira mitochondria membrane oira chloroplast ki membrane oira whatever membrane haba wai si wai ya hairak pataragadi membrane khuding mak ki structures it is explained by fluid mosaic model and explained there and membrane is made about how many lipid layer so lipid bilayer ani yawe hydra ana sir na sida isa lipid ki bilayer somna kanai ma somna kanai smile layer mani bro aduna ne membrane kya khatana adi chloroplast lipid bilayer kya yomne chloroplast mari yomni because it is a double membrane na membrane amamda lipid ki layer ani membrane ani dadi mari yawroidro ade membrane am khatane have a sarni se thylakoid is a single membrane bounded structure ne yare ko manung se thylakoid ki manung se hangeba sigi manung space se we call it lumen haina ko kai ko ba se this is called lumen yare ko now, sige membrane se sir, kitang kag zoom out taro kiyo ga adu kanda understand taro ni. Adu di sir na si thylakoid ki membrane dune laura sine. Yare ko? Kay na saram ni ba lipid na? Chumi ko. Maram di ako'y na cell biology dagi kangi basi plasma membrane is made up lipid bilayer na sari hai. And also tamapada bilayer sing bi marak ta proteins mayam so embed taro yawi hai ki. Hai ki ko? Ana ikaram ta hapsan ge. Liyeng si ko mayam. Loy ma yek to inati matiyam katang palam ge to gani. Kanagi membrane sarnigi se. Hariba thylakoid ki membrane singsida. Within the membrane sida na. Pigments are embedded in group ko na layer. Yana, kana pigment, chlorophyll A, chlorophyll B, carotenoids, xanthophyll, chlorophyll C, chlorophyll D, all the pigments are grouped together. Yani ko, yung ko aduri ay silam tayo ko, kaya wala group ko na layer ba? Thylakoid ki membrane ki lipid bilayer mark to the pumalayer. Asumay na layriba pigment sing sige group se we'll call it photosystem ko we harap to neba hai ting aida amtang tagi si kaya wai dano lay si pigment se onto the thylakoid membrane right so this is pigments this is another pigments this is another pigments this is another pigments this is another one so ang pigments group wai layer kaya wai dano si group se lipid bilayer mark to the yare ko so what the pigment sing se group wai na lay hai tari moy mark to glue na phada di yaro ne room na phada di ko chai chare lei roidra. They must be held together, right? Kana na phage the protein na phage. Homegrown sir na say, what is photosystem? Photosystem is a group of pigments together with the protein. Protein joy of photosystem high tardy. Yarabo. Aduri ngasai ko tunnel protein guy na yaw mayam do. To aduga dum pullaga lay grouping ne pigment sing siga. Yaraba. Aduna ako'y kamay define tawni intrinsic extrinsic proteins kan yaw gidana. Ano protein do dum associate to lay group of pigments ni na harak to ne. Aduna we better define photosystem as kay group of what? Group of pigments associated with proteins. So proteins ka associate to lewa. Group of pigments associated kanaga with proteins yawi found located on kaiwaida thylakoid membrane. Found embedded on hydrosui found located on thylakoid T membrane sing the napagalito net thylakoid or grena hydrosui ko thylakoid membrane. Moishibo kina kay ko ba photosystem haina ko. Yarabro sarnai se yari ko maya ana horene kitang controversial oidana ba ay kinang taga hayoge ka. Sani si kanay lipid baile na. Thylakoid ki adugi marakta pigments in group oina lay together with the proteins we call it photosystem. Hariba lipid bilayer marak sida si kakta na tana kanasule. We have different electron carriers mayam sulay kambiyo. Yani ko si bilayer marak sida kay mayam sila electron pura ka chatpang ba carrier mayam sila. Horen na light reaction ta pa nungay na. Hangro. Yani ko. Okay. Shall I rub this? Silaw thokige nungay ti. Hay taba mali. Noy di hay rena. Yani ko. Yaraba? Okay, other tardi hoji question am tholak tore. Atay loimak samdatlagasu auto system ek palakpada group ne haibam dikhang menski hairiba group se combine organized toge yani ka the group is organized in such a manner that at the center of the group there should always be chlorophyll A lane yani ka grouping se sema matamda group tu ki mayai da kana leita yadeba chlorophyll A layer the surrounding pigments can be any other pigments ato pigments no be yai hairak to ne yare ko aduna aikoina photo system ama yekpa matamda sumaina yekani always at the center of the group what should be there there should be chlorophyll A group of pigments anina pigment mayam na pula sa donina pigment mayam na punba du da mayai do ina kai pigment di hapta yadaba chlorophyll A happy hairiba chlorophyll A sige akoi bada there can be any other pigments atopa pigments yawa yai yare ko so these are the other pigments it could be carotenoid xanthophyll chlorophyll B chlorophyll C chlorophyll D oiba yai haiba okay so, so what am I doing what am I doing I'm drawing a photosystem. Photosystem amagi mayayda layriba chlorophyll A se we call it reaction center haina ko. Horen siday ni reaction ho doise. Kay ko ba se we call it reaction center. Yari hayo reaction center se nataba lemori ba pigments sing se the remaining pigments are called accessory pigments. Accessory ID yaurasu yaba yaudasu yaba poche. Yani ko adi photosynthesis tangai phadana tawge adi kai di yaudasu yadre. Chlorophyll A the yade. I don't know whether you remember it or not. Chlorophyll A is called universal pigment. We discussed about three types of algae, right? Green algae, brown algae, and red algae. Humuk takay lo yawe. Chlorophyll A, chlorophyll C kana yawe. Brown kanada. Chlorophyll A na B na kanada, green algae da. C na brown algae da. D na kanada, red algae da. Yarni ko. Now question is green algae da chlorophyll C yawe. Yauwde to adi C's di yarswe yauwde sweve. D's di yarswe yauwde sweve. But yauwde ba yada bam di leve kana. So main, the most important and major pigment sidi kana no chlorophyll A because the reaction will start from that pigment. Hence it is called kai koeva reaction center koeva. While the surrounding pigments are called kai koeva, we call it accessory pigments. Haine koeva. Moishina kai koeva accessory pigments koi. Yare ko. Remember, accessory pigments are also called antenna molecules. Kay ba? Antenna molecules. Yaribro mayam si faubada. Yareko. Kadino duri sir ni isa. I'm drawing the general structure of a photosystem. Amana laibham thokpa structure mong dune. Yareko. Shall I proceed? Okay. Now, photosystems makal ani thokna khaidokpa yae. Yareko. We have two types of photosystem. One is called photosystem 1 and another is called photosystem 2. Amana photosystem 1 haibama, amana photosystem 2 haibama. Kaida khena gengsi. Somdo intake. Okay. Now, look here. Types of photosystem. Makoi karam kande hogibana class kai thade hogibana. Edition kai thade hogibano. Sa lyric paring aida aduga loi kawhreda kam hairo. Kaino karam kande hogige khoi. Kaide. O loire class ho timing kawlen. Hoji fauta tha kya surage ba khoin talise. Tau maning da changsalak toise. Siga akoina lansalation yam ti hele ba lansalation. Yare kwa lansalak pai si yamna lana lansalak panate nam imanaba imanabi mayamna koi ba chatna rakanda akhoi test ki kanna paba mayam su yau hi. Yare kwa haidi sacrifice kharadi tawrure ko sacrifice kharadi tawthure aduga marak sida hek give up tawgi tardi nang na hange tawri do kyam ganar roiba ko aduna hojik sina kitang thap amaga tawbiro kitang yam ga hai roi ai machet am tanga yare ko kai khobi du budi sacrifice tawthukre khudom bi dum tanga tawrdi yarni photo system si makalani lay we have two type of photo system. One is called photosystem 1, another is called photosystem 2. Munatasi, si kangbada ang kayam loiramni light reaction. Yare ko. Amana kay koiba, amana ne photosystem 1 koiba. Ayna noida haige to ise atay natira. Maram tang thalaw ko sir. Kitang di pressure laibane ba sige exam sini. Yare ko. Radip kasina laiba pressure si lore matam amada. Maso medical tharmine. Ki laibane. Ko mangda sudum physics ko hotpado. Sigumba se am tawni ba sidi paikha do loidri bokti khara. Kaiginoda yamna nong nini khaki preparation na tamnina marakta to nahan gidumak sumangai testa yam phangta matang jawni yam phang patang jaw aduda hekta test amna decide tawdam na te ba sarna ise aigi nungtigi pung manga taruk taruk papam thokpada dum lengdana pabada handar oiba kashu ko papam thokpadu nang pawa paba handara kanda ne ba handa doise ma karam ka handara kaniya paba handara kanda yani ko ma yam phangdra kanda mathangi hai handaki pekhada ma yam phangodreba mathang pekhada ma handara kadam na tibaduna yani ko ma handara kada factor di paba handara kanda pabadi suwa handadriba adum khonariba miduna di suwa handaroi habani sarna isana paba lep kano yani ko one is photosystem 1 another is photosystem 2 photosystem 2 is also called PS2 sugoi yare somdengo kaisu ko eba PS2 asambida PS1 PS2 haise kaigi matungina maming tholino haitari in order of discovery ne adi miyoibana panbida photosystem ani leirise kanana hana khangba horakige haitardi kanana photosystem 1 hana khangba horaki haineba yare ko horin NCR the even ko the names the Roman numbers are not given not based on this order of reaction photosystem 1 dagi hana reaction hobai na teba yarni ko horin reaction di photosystem 2 de horkani khangi ko aduna sida 1 2 haina tholise in order of kaida so photosystem are given the name 1 and 2 based on their order of discovery da yare ko aduma ai isan badadi hap kong ge ko sir photosystem 1 se ai di maming amasu thonjenge it is also called P700 so ko kai ko ba it is also called P700 while photosystem 2 is also called P680 so ko yari hayo P680. ꯌꯥꯔꯦꯀꯣ? ꯑꯗꯨꯗꯤ ꯁꯔ ꯑꯅꯤꯃꯛꯀꯤ ꯃꯌꯥꯏꯗ ꯀꯃꯟ ꯑꯝꯇ ꯂꯧꯒꯦ. ꯗꯥꯏꯒ꯭ꯔꯥꯝꯁꯤ ꯐꯣꯇꯣ ꯁꯤꯁꯇꯦꯝ ꯑꯃꯒꯤ ꯃꯌꯥꯏꯗ ꯃꯇꯝ ꯂꯩꯕ ꯈꯤꯕꯤꯛꯇ ꯀꯩ ꯂꯩꯅꯤꯕ? Chlorophyll A ꯂꯩꯒꯅꯤ. We call it as reaction center. ꯌꯥꯔꯦꯀꯣ? ꯍꯥꯏꯔꯤꯕ reaction center ꯁꯤꯒꯤ ꯑꯀꯣꯏꯕꯗ ꯑꯇꯣꯞꯄ pigments ꯃꯌꯥꯝ ꯑꯃꯅ surround ꯇꯧꯏ. Surround ꯇꯧꯔꯤꯕ pigments ꯁꯤꯡꯁꯤ we have given the name accessory pigments. Now one question arises. Question ꯑꯃ ꯊꯣꯛꯂꯛꯑꯦ. ꯌꯥꯔꯦꯀꯣ? ꯀꯩꯗꯧꯕꯒꯤ reaction center ꯁꯤ pigments ꯅ surround ꯇꯧꯏꯅꯣꯅ ꯑꯩ ꯉꯁꯥꯏ answer ꯑꯃꯗꯤ ꯍꯥꯏꯈ꯭ꯔꯕꯅꯦ. ꯀꯔꯤꯒꯨꯝꯕ ꯑꯩꯁꯦ ꯁꯔ ꯁꯤꯗ ꯏꯊꯟ ꯂꯦꯞꯄꯝꯃꯕ ꯑꯗꯩ I will be directly exposed to light ꯌꯥꯝ ꯊꯤ ꯁꯥꯔꯅꯤ ꯑꯗꯨꯗꯤ direct hit ꯇꯧ ꯇꯥꯔꯗꯤ photo oxidize ꯇꯧꯔꯒ chlorophyll pigment ꯁꯦ damage ꯇꯧꯕ ꯌꯥꯏꯑꯕ ꯑꯗꯨ protect ꯇꯧꯅꯕ ꯑꯃꯅꯅꯦ in order to absorb different wavelength of light ꯁꯦ absorb ꯇꯧꯕꯗ efficient ꯑꯣꯏꯅꯕꯅꯦꯕ I will let you know ꯑꯩ ꯍꯧꯖꯤꯛꯀꯤ ꯂꯥꯏꯟꯗꯣ ꯀꯩ ꯍꯥꯏꯅꯣ ꯍꯥꯏꯔꯛꯀꯦ ꯉꯥꯛꯇꯪ ꯉꯥꯏꯈꯣꯀꯣ. For example ꯃꯨꯟꯅ ꯇꯥꯎꯀꯣ ꯁꯔꯅ ꯑꯩꯅ absorb ꯇꯧꯕ ꯉꯝꯕꯁꯦ ꯀꯣ ꯑꯩꯅ absorb ꯇꯧꯕ ꯉꯝꯕ ꯑꯩꯒꯤ maximum capacity ꯁꯦ ꯑꯩꯅ ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯄꯥꯝꯖꯅ absorb ꯇꯧꯕ wavelength ꯁꯦ for example 600 ꯅꯦ ꯂꯧꯔꯁꯦ 600 nanometer ꯌꯥꯔꯦꯀꯣ ꯁ꯭ꯋꯥꯏꯗꯅ 400 ꯈꯛ available ꯑꯣꯏ ꯂꯩꯔꯦ ꯌꯥꯔꯦꯀꯣ ꯑꯗꯨ ꯑꯣꯏꯇꯥꯔꯗꯤ ꯑꯩꯅ absorb ꯇꯧꯕ ꯉꯝꯈꯤꯗ꯭ꯔꯥ ꯃꯔꯝꯗꯤ ꯑꯩꯅ absorb ꯇꯧꯕ ꯉꯝꯕ ꯑꯩꯒꯤ wavelength ꯁꯤꯗꯤ ꯀꯌꯥꯅꯣ 600 ꯅꯦ available ꯑꯣꯏꯁꯤꯅ ꯀꯌꯥ ꯑꯗꯨꯅ 400 ꯇꯣ absorb ꯇꯧꯕ ꯉꯝꯕ accessory pigments ꯑꯃ ꯊꯥꯗꯣꯛꯄ ꯑꯗꯨ ꯃꯥꯅ ꯑꯣꯏꯅ collect ꯇꯧꯔꯅꯤꯀꯣ 400 ꯊꯪꯅꯕꯗꯨꯅꯁꯨ 400 ꯄꯨꯂꯞ channeling ꯇꯧꯔ ꯌꯥꯝꯈꯠꯍꯜꯂꯀꯥꯟꯗ ꯑꯣꯏꯅ ꯊꯥꯗꯔꯛꯄ ꯑꯩꯉꯣꯟꯗ tunneling effect ꯍꯥꯏꯇꯥꯔꯦ I will show you here ꯌꯦꯡꯉꯨꯀꯣ ꯁꯤꯗ chlorophyll surrounding pigment orange ꯁꯦ ꯀꯩꯅꯣ accessory pigments ꯁꯤꯅ ꯍꯥꯟꯅ collect ꯇꯧꯒꯅꯤ ꯍꯥꯏꯕ light of different wavelengths ꯌꯥꯔꯅꯤꯀꯣ ꯃꯊꯪ pass ꯇꯧꯔꯀꯥꯟꯗ finally ꯀꯥꯏꯗ ꯊꯥꯗꯗꯧꯕ chlorophyll air so they are funneling ꯌꯥꯔꯦꯀꯣ the entire energy into the reaction center ꯗ ꯊꯥꯗꯗꯣꯏꯅꯦ ꯌꯥꯔꯦꯀꯣ ꯈꯪꯉꯤꯕꯣ ꯈꯪꯉꯤꯀꯣ so what is this what is coming here light ꯑꯩ understand ꯇꯧꯔꯤ ꯍꯥꯏꯅ ꯂꯧꯔꯒꯦ ꯃꯌꯥꯏꯒꯤꯁꯦ ꯀꯩꯅꯦꯕ ꯃꯌꯥꯏꯒꯤꯁꯦ ꯑꯇꯩ ꯏꯗꯁꯨ ꯃꯌꯥꯏꯒꯤꯁꯤꯇꯪ 이게 sir central pigment is called reaction center 하이나 कौवेबा यारेको हौजिक सरना हाइरक्तोइसे photo system से मखल अनि लै कईदा बेस तौर खाइदोइनो हाइतारदी photo system दुगी मयाइदे याउरिबा reaction center दुगी absorbing capacity दा बेस तौरगा करिगुम्बा reaction center सिगी मागी maximum wavelength absorb capacity से 700 nanometer wavelength चुपसन बंगमबा हाइतारदी we call it photo system 1 का 680 भोक्तंग अमबा somewhere 600 to 680 swai भोक्तंग we call it photo system 2 को adi longer wavelength absorb tawma misa karamba photo system na photo system 1 shorter wavelength absorb tawma misina kanana no absorb toise kana gi wari no reaction center wari ni ko accessory pigments nati ko kari gumba reaction center sina if it absorbs wavelength of light 700 nanometer we call it photo system 1 if it absorbs 600 to 680 nanometer bhopodang hadi shorter wavelength when absorb tawma magana we call it photo system 2 haina ko bro khangiko okay ꯅꯪꯁꯨ ꯑꯩꯁꯨ human species ꯉꯥꯛꯇꯅꯦꯕ ꯌꯥꯔꯅꯤꯀꯣ but ꯑꯩꯅ ꯇꯧꯕ ꯉꯝꯕ ꯀꯦꯄꯥꯁꯤꯇꯤꯒ ꯅꯪꯅ ꯇꯧꯕ ꯉꯝꯕ ꯀꯦꯄꯥꯁꯤꯇꯤꯒ ꯈꯤꯇꯪ ꯈꯦꯠꯅꯔꯣꯏꯗ꯭ꯔꯥ ꯑꯗꯨꯒꯤ ꯀꯦꯄꯥꯁꯤꯇꯤ ꯈꯦꯠꯅꯕꯒꯤ ꯃꯇꯨꯡ ꯏꯟꯅ ꯇꯣꯉꯥꯟ ꯃꯃꯤꯡ ꯊꯣꯟꯕꯅꯤꯗ ꯌꯥꯔꯅꯤꯀꯣ ꯀ꯭ꯂꯣꯔꯣꯐꯤꯜ A ꯁꯣꯝ ꯁꯥꯏꯠꯀꯤꯁꯤꯅ ꯃꯥꯅ ꯆꯨꯞꯁꯟꯕ ꯉꯝꯕꯁꯦ ꯀꯌꯥ ꯆꯨꯞꯁꯟꯕ ꯉꯝꯕ 700 nanometer wavelength ꯁꯦ ꯌꯥꯝ ꯄꯥꯝꯖꯅ ꯆꯨꯞꯁꯩꯌꯦꯕ ꯁꯣꯝ ꯅꯥꯀꯟꯗꯅ ꯀꯌꯥ ꯐꯥꯎꯕꯗ 680 shorter wavelength ꯆꯨꯞꯁꯜꯂꯒꯅ photo system 2 ꯀꯧꯏ ꯍꯥꯏꯕꯅꯦꯕ ꯑꯗꯨꯗꯤ ꯅꯣꯏꯅ ꯁꯤꯗ ꯍꯪꯉꯛꯄ ꯌꯥꯏꯕ ꯀꯩꯒꯤꯅꯣ ꯁꯔ ꯀ꯭ꯂꯣꯔꯣꯐꯤꯜꯗꯤ ꯀ꯭ꯂꯣꯔꯣꯐꯤꯜ A ꯉꯥꯛꯇꯅꯤ wavelength absorbing capacity ꯈꯦꯠꯅꯩꯁꯦ ꯀꯩꯒꯤꯅꯣꯗꯤ ꯑꯁꯦꯡꯕꯗꯤ ꯀ꯭ꯂꯣꯔꯣꯐꯤꯜ ꯁꯤꯗ ꯅꯠꯇꯦꯕ ꯈꯦꯠꯅꯩꯁꯦ ꯉꯁꯥꯏ ꯐꯣꯇꯣ ꯁꯤꯁꯇꯦꯝꯗꯤ ꯀꯅꯥꯒ ꯑꯦꯁꯣꯁꯤꯌꯦꯠ ꯇꯧꯋꯦ ꯇꯣ ꯃꯥꯒꯤ ꯑꯀꯣꯏꯕꯗ ꯂꯩꯔꯤꯕ ꯄ꯭ꯔꯣꯇꯤꯟꯁꯇꯣ ꯃꯣꯏꯒꯤ ꯕꯤꯍꯦꯚꯤꯌꯔ ꯍꯣꯡꯕꯒꯤ ꯃꯇꯨꯡ ꯏꯟꯅꯅꯦꯕ ꯀꯦꯄꯥꯁꯤꯇꯤꯗ ꯈꯤꯇꯪ ꯏꯝꯄꯦꯛ ꯂꯥꯛꯏꯁꯦ ꯌꯥꯔꯦꯀꯣ ꯑꯗꯨꯃꯥꯏꯅ ꯌꯥꯝ ꯗꯤꯞ ꯑꯣꯏꯁꯜꯂꯒꯨꯝꯁꯤ ꯑꯩꯈꯣꯏꯗꯤ ꯌꯥꯝ ꯁꯤꯝꯄꯜ ꯑꯣꯏꯅ ꯐꯣꯇꯣ ꯁꯤꯁꯇꯦꯝ ꯍꯥꯏꯇꯥꯔꯗꯤ ꯀꯩꯑꯕ ꯍꯤꯌꯔ ꯐꯣꯇꯣ ꯁꯤꯁꯇꯦꯝ 1 ꯑꯗꯤ ꯀꯩꯑꯕ ꯍꯤꯌꯔ chlorophyll A reaction center has absorption peak kayabuk chupsan mga meba has absorption peak at mana pamjana khwaidagi peak yauna chupsan mga mase peak at kyada 700 nanometer bok chupsan mga ngani yare ko somna kandana kaya here chlorophyll A absorbs I'm talking about chlorophyll A the reaction center ni ko absorbs Web length kya? Web length of 680 nanometer vok chupsalagana, we call it photosystem 2 haina khangne. Yari bro mayam. Yari ko. Now, amatanga hapam dore sina sir roive explanation su oidromni munatabi roko. Ngasaya koi chloroplast check ko sir. Si chloroplast sir amtang copy to pa yabre nge sir. Can I change the slide? Ay sir handakti NCRT da khaktang khak base tawni ko. Word me am di yawe yabra amtang check tawge. Yes, yare. Okay, look here. Yung ko sila. Hojik sir na sila lairisi ay manungisi ngasay su gyan tagrhamnina muthatkrage. Manungsi kai koi swaisa. Stroma purple machu singsina kai thylakoid. Thylakoid maron longkalisina kai. So this is one grana. I will draw another one. Another one thylakoid, 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 thylakoid. Yareko. This is another grana. Hangnaba grana ani se they are connected stroma moigi thylakoid ani se samnayeva. Kai kawenta mai se we call it stroma lamelli haina ko. Stroma kai ko ba se lamelli. Swaida kitang khak muna taw ko. Ngasidi live ningni hayeng sarna non cyclic photophosphorylation kai da chathai cyclic photophosphorylation kai da chathai haiba kanda swai gi concept se kitang sengaudari prom khar laini. Photosystem kaya lai kai da laini ba thylakoid ki membrane da hariba photosystem ani shira photosystem 1 se photosystem 1 se kai wai da lege hamtam da photosystem 1 is located on the grana and also on the stroma lamelli ani maktale kai da leba photosystem 1 se photosystem 1 is found located on the stroma lamelli as well as the granule thylakoid ani maktale ni yare ko aduna sir horin ani se comparison tawrap na piba yanaba photosystem 1 is found located on kaida layeba madi located on stroma hiro stroma nattaba grana de hoge located on grana da sulai and kaida sulai and stroma lamelli de sulai mayam ya bro sir na hairi sibo stroma lamelli sida yellow keno ngakna pisan ba nungaitre haragana sir se machu amna ibige se to sigi aine uliba orange machu se kai neba this is photosystem 1 neba yare ko adi photosystem 1 se akhoi grena da su ure photosystem 1 se akhoi stroma lamelli de su leiba ure yare ba sumaina adumai to dana photosystem 2 gi system se sina kadai waida leige haitari they are found located only on the grena khakta da le kai da leba so sir na sida ping na uli sina kay tare photosystem 2 so they are absent in stroma lamini adai sir kanam ta have ko ka lagam bok lak ba na tam ni na hayeng mapa du da ay am details ta ke but sum khang ba yaw ring se adi grenada photosystem ani mak leire ne ps1 so ps2 so an horen grenada non cyclic photophosphorylation sa thai her kani because non cyclic high river process sa thagi ay tari photosystem ani involved stroma lamini dedi kai system ka yawi sidana horen cyclic photophosphorylation sa thai na harakani haba yare ko hana khangdrasu yare dai ko na hairage asom site na kai da yawewa located on grena sina kai da yawdaba grena da khaktam yawa adudi grena da leise photosystem khu kya ne ba ani 1 su lei 2 su lei lameli da leise stroma lameli da leise only to 1 khaktani haina mai phang tare sibokta ai photosystem se understand tore hana lo yaraba yarage ko shall we directly start from the mechanism tomorrow sibokta thank you